areas. Yeah, just a message that the meeting will be recorded, so I hope everybody is okay. Thank you for joining again. I think some people join every week, which is great. Some people are new and they haven't maybe seen all of us. So I'm Stephanie Reichelt. I have been until last week head of light microscopy at Cancer Research UK, and I'm now actually joining the Sanger Wellcome Institute as strategy science strategy manager for the Human Cell Atlas. So this is hopefully not making much difference for the Imaging One World lecture series, but it's just making maybe a difference to my life. We have Heba, Sailem today, and which I met actually. She was one of the last speakers in Cambridge just before lockdown last year. That's when we met, which was very awkward because I think we were just getting used to nobody being able to talk to anybody else without masks and not through Zoom. But so it's a pleasure to see her today in a Zoom meeting presenting her wonderful work. And she'll be introduced by Nick Barry from the LMB. And I'm just kind of preparing the final quiz questions so you can have a full scope quiz at the end of this. So I hope you enjoy.
Hi, it's a real pleasure to introduce Heba Sailem. Heba is currently a Henry Wellcome Fellow at Oxford University, basically working on this very difficult problem of how you visualize data that you extract from one media, such as microscopy, alongside data that is coming from, say, genetic work and what have you. So it's a very interesting area of work. I guess in your PhD work, you came out with this really interesting idea of linking the cell shape and morphological properties into a sort of more complex and overarching view of all the data of the Phenoplot. Really nice piece of work, excellent. And I mean, nowadays, I guess a lot of your work is actually application of these sort of approaches to different problems, such as in cancer research and what have you. I guess really, I'll just let you get on and tell us all about your work. So I'm really looking forward to it. Thank you very much, Heba.
Thank you very much, Nick, for the introduction, and thank you all for tuning in. So I'm a research fellow at the University of Oxford, working between the Big Data Institute and the Institute of Biomedical Engineering. And today I will talk about some of the technologies I developed for analyzing large-scale imaging data, as well as translating these very rich data sets into a biological knowledge, which is always the hardest task to deal with. And so far, my focus has been on understanding tissue architecture, and multicellular organization. And this is very important in cancer progression because cells that organize in glandular structures, as you see here, for example, start to change when cancer occur. And in low-grade breast cancer, we can see some changes, but as cancer progresses, we see that the cells lose their architecture and their organization. And this is basically, they don't become any more synced with each other. And over the last decade, we have done an amazing job at characterizing genetic alterations that result in these changes in tissues that associate with cancer initiation and progression. However, what is maybe wasn't as much appreciated, or just becoming more appreciated now, is that when the cells start losing their organization and the tissue architecture change, this exposes cells to more physical forces, to different mechanical cues that also drive signaling via mechanosensing. And it's really important to understand cancer is to study all these factors together. And of course, imaging is the perfect approach to look at what drives tissue organization and tissue morphology. So the basic component that shape the tissue is at the end is start from the cell, so cell morphology and shape, which is an outcome of many different interacting components. So we have the cytoskeleton like microtubules or actin filaments. We have cell-to-cell adhesion and cell-to-matrix adhesion. And all these factors work together to give rise to the cell structure and tissue structure as well. And cell shape has been for a long time used as a proxy for the physiological state of the cell, like cell division, apoptosis, differentiation, or migration. So it's quite rich readout to be used in many biological assays. However, cell shape is not only a passive readout, it's also play an active role in cell signaling. And I like always to give this example that by Kilian et al., who show that actually by changing the shape of the cell, you can change its fate. So here we have mesenchymal stem cells cultured on a different islands, like shaped islands, so a circle or a bipolar shape. And just by the cells having different shape result in a different differentiation outcome. So cells cultured or have like a circle shape are more likely to differentiate in adipocytes, while bipolar cell shape cells are more likely to differentiate in osteoblasts. So here we are not changing the genetics, just changing the shape, and we completely have different outcome, which highlight why it's important to study these components. Of course, this is not only for stem cells. So my own work during my PhD, I also showed that by statistical modeling of thousands of measurements from imaging data, that NF-κB activation, which is an important transcription factor, also can be modulated by various aspects like cell context or cell-to-cell contact, protrusions, and geometry. So even in a homogeneous population, depending on the context of the cell, we can have different activation of or different signaling outcomes. So today I will talk about different ways we can study the context, the organization of the cell, and the shape of the cell by carefully designing features that can inform us about these aspects. I also will talk about the method I developed for automatically and systematically studying genes involved in multicellular organization using genetic screens. And I will end up with an update on Phenoplot project for visualizing microscopy data. So the first example is, you will see from this talk that I'm using very simple data sets now. There is multiplexing and tissue imaging, which is far more complex, but even from simple data, we still have a lot to achieve. So usually with wound healing assays, we have like induce a wound, a scratch, like artificially, and then observe how much the cells will close the wound over a period of time. So here, 24 hours, for example. And when biologists do these experiments, which is a widely used tool, they only look at how much the wound closes. So for example, here, for this control condition, we have a certain closure of the wound. However, after knocking down genes involved in motility, like CDH5 and CDC42, we observe significant reduction on how much the wound closes. And this reflects that they have a role in motility. However, if we look at the effect of these knockdowns, we see that they actually have very different effects on the cell organization, on the morphologies, etc. But the challenge is, if we want to do these experiments like in a flexible way, these images were acquired at 4x and acquired for thousands of conditions. So we want a flexible approach that allow the biologists to do what they do normally, and then come up with analysis approach that allow them extracting more information from these data sets. So what I've done is to develop a deep learning approach that allow detecting cells in these images. So by for first annotating the location of different cells, we can train a neural network. So here, like inspired by unit architecture, which we call it here deep scratch, is to predict the location of the cells based on these annotations. And you see here is the location of each cell is basically represented by a Gaussian, so probability, so it is not like the error, the banalization or the error will be less if it's the prediction is within this range. And what is nice about deep learning is that we're not restricted to certain markers. So in this data set, we have images with this cytoplasmic marker, and we have a membrane marker. And we trained both of them, like the network will take input both of these images and predict the locations very robustly, as you can see. Once we learn this representation, then the biologist can apply the network to learn from any set of markers, which is very useful and very flexible. So once we learn the locations of the cells, I applied Voronoi tessellation here, and basically it provide approximate segmentation of the cells. So the pixels around the center that closest to the cell center would be assigned to that center. And in this way, we have this like a way to approximate the cell area and how much density of the cells around. Using this like local cell density, like the distance to nearest 6 neighbors or 36 neighbors, like how many layers of cells, then and normalizing this to the distance to the wound center, I showed that actually now we can separate different conditions much better. So the mock samples now are separated from CDH5 and CDC42 knockdowns, which basically tell us more information about that actually these conditions or these knockdowns can result in is perturbing motility or affecting motility through different mechanisms. And we know CDH5 affects cell adhesion, while CDC42 affect the polarity of the cells. Another example that going up level, like the tissue scale properties. So here in this image, we have endothelial cells cultured on a matrigel, and these cells have an amazing ability that once they cultured on a matrigel, they even in vitro, they start self-organizing and forming these capillary-like networks, which make it very flexible tools to do like various perturbation and study the effects of various perturbations. So here, for example, we see the effect of different drug treatments, and you see here, like even the tubes have different properties. So what basically usually biologists do is they measure the total length of the tube. So just segmenting the tubes and measuring that, which doesn't give us a lot of information about all the aspects in the network that also can provide a finer details on drug effects or genetic perturbation effects. So what I developed is an image analysis algorithm that measure like more than 100 features from these networks by segmenting the different elements in the network, like tubes, nodes, and meshes. And then from these different elements, we can calculate different statistics like the mean, the total, the standard deviation. We also can use concepts from graph theory to look at how much efficient hypothetical flow would be. So for example, by measuring the distance between X and Y, the shortest path, we can do this for every 2 nodes and average that over the whole network to obtain a robust measure of how much efficient the flow in this network would be. Another aspect we can look is at the orientation of different branches to see if they are like binding by certain biophysical rules. So all these measurements can provide us with more comprehensive characterization of treatment effects. So I applied this to data set that was generated using high content screening of 1,028 drugs using the low-pack drug library. And from the 100 features, we applied Disney, and you can see here now, we're using clustering, we identify different signatures for these different networks. So here you see the control condition, here drugs that inhibit formation of tubes, while in this cluster, we see the like thin and the small branches, and while here is the more like shaggy branches, not very mature. And this cluster in the purple is resulting in improved network formation. And what's interesting that I was able to validate by integrating with other gene expression data from Alzheimer's patient that actually some of the drugs that target glutamate receptors actually affect angiogenesis and affect patients' outcome. So showing that actually these phenotypes that we characterize in vitro are relevant. So the second part I would like to talk about is analyzing genetic screens. And with genetic screens, we show like a lot of work shown that we can use CRISPR knockouts or siRNA knockdowns to infer gene functions by observing how the phenotype of the cells change after knocking down the genes. So here we have cells like in blue, the nuclei in blue is in reflecting the like Dabi stain, and the green cells are reflecting rotavirus infection, which was the interest of in that data set. So what we can do once we knock down every single gene, then we see how it changed the phenotype of these cells. So if the cells become more round, then this can reflect a role in contractility. If the cells become large in compared to the control cells, then we infer a role in aging and senescence. And we can do this for every single gene in the human cell. And once we identify genes that have similar phenotypes, so here gene 2 and 4, really large cells, we can say they are functionally related. However, the analysis of these data sets, once we start doing genome-wide screens like 20,000 gene knockdown, become very challenging. And people who started doing these screens started with big hopes. But at the end, they were only able to focus on one feature because we couldn't make sense of the entire spectrum of the features we're deriving from these screens. And one of the challenges of that is the heterogeneity. So I'm showing this small crop of the image, but actually what we are measuring is thousands of cells. So here is just one image having like around 1,000 to 2,000 cells, and we have 4 images like that for every single gene. So there is a lot of heterogeneity in terms of local cell density, in terms of cell shape, and even how much rotavirus infection happen in the well. So also we need effective ways to deal with this heterogeneity. So basically what I wanted to do, which was the part that I proposed for the Wellcome Trust, is to develop a systematic way to analyze these screens. And to do that, I collaborated with Lucas Pelkman's lab, who have like a data set, like a siRNA screen in HCT116 cell line. They have performed around 18,000 gene knockdown. And for every gene, they image the entire well, which give us a lot of information on cell morphology as well as how cells organized in the well. So overall, this data set resulted in like 500 million cells, which is a huge amount of information to really look at these different aspects. In the data, we have 2 markers, DAPI and viral protein 6, which is detected by rotavirus infection. So basically, we perform image analysis, so segmenting every single nuclei and cells and measuring various features like texture, DNA content, cell cycle, etc. We also measure the microenvironment, so whether the cells at the edge of the colony or not, and the local cell density. So cells in the middle of the colony have higher density, while toward the edge of the colony they have lower density. other features like different cellular states like apoptotic, mitotic cells, or if the cells are infected or not. So from these measurements, we have for every cell 160 features, and for every gene knockdown, we have around 6,000 cells. So the question how we will represent this to get the gene profiles. And what I've done here is like to use different statistical measures to try to capture the heterogeneity and if the gene have a pleiotropic effect. So sometimes genes have different functions, so they will result in one phenotype in one subpopulation, another phenotype in another subpopulation. And I wanted something that is very flexible and can be applied systematically without like a lot of customization. So what we done here is like computing the mean, like the average, but also other quantiles. So the quantiles reflect the presence of subpopulation. So the lowest 5%, the highest 5%, the variability, so the interquantile range, as well as different features of the distribution. So like skewness, biomotility, what is the different distance between the control population and the perturbed population. So basically from this, we converted 160 features per cell to 1,700 features per gene. And this is like, we hope that this will represent the various phenotypes that the gene is resulting in. So then become the question, okay, we have the genes that we know we perturbed, and we have measurements that reflect the phenotypes that resulted from the perturbation, and how we can convert this to knowledge about what are the functions that the genes doing. So my idea is to start from what we know. So for example, if we know that a certain set of genes play a role in cell adhesion, then we can use these genes and search using machine learning for phenotypes that associated with perturbing these genes. And once we identified these genes, the phenotype associated with cell adhesion, then we can find other genes that have a similar phenotype and also predict that these genes have a cell adhesion function. So this is like a systematic way like to link genes to phenotypes and functions. And I call this approach that we start from what we know to discover phenotypes, knowledge-driven machine learning. And the question is where we will get this prior knowledge, because in any experiment, we don't know a priori what are the genes that involved in cell adhesion. So what I used information from gene ontology, which is like a reasonable starting point. And here what I do is to retrieve genes we know that they involved in cell adhesion, and try to find if there is a subset of the phenotypic features we have that distinguish them from a random set of genes. So if we identify such subset, then basically simply what we can do is to train a classifier that distinguishes between cell adhesion genes and other genes. And this classifier then can be applied to the entire data set to predict which genes are positive, so in orange, the positive ones, and based on the phenotypic similarity, also give it a confidence score of these predictions. So I think this is here a very important idea that actually why then we don't take all the features and see how the cell adhesion genes how they affect all the features. However, the problem is that perturbing cell adhesion might affect only subset of features. So if we take all the features together, basically we are losing the signal. So here, the cell adhesion genes in reduced dimensional space of all features, we don't see a signal, we don't see a signature. However, once we take only the subset of features that seem to be associated with cell adhesion genes, then we are more able to deconvolve the signal from the high dimensional space and guide the machine learning in a more efficient way. And this is like a way to handle the high dimensionality of these types of data, as well as the low signal that can be lost when we consider everything together at once. So we can apply this approach then to any gene function. So based on gene ontology, I selected genes that gene ontology terms that have between 100 genes to 500. So not very general, not very specific. And then we apply this to learn the phenotypes associated with these different functions and basically have multiple ranking for each gene. So this is how it's look like. For example, we have a cell adhesion classifier that identify genes associated with cell adhesion based on phenotypic similarity. Then maybe based on our markers, there is no phenotype associated with hormone activity. So if the classifier doesn't have a certain performance, we don't use it. And we do this for many functions, and this result in multiple ranking for each gene. We have multiple predictions what the genes are doing. And of course, along their confidence in these predictions. So I call this approach knowledge and context-driven machine learning. So it's driven by knowledge, as I said, but it's also context-driven because it is the predictions we will have, it will be based on the data that we have. So for example, if we're looking at the colorectal tissue, we might predict different function if we're looking at breast tissue or other tissue types. So this is very important because currently gene ontology doesn't offer that. And by having this way systematically, we basically start inferring tissue-specific or disease-specific gene functions. So how this can be used? So for example, I'm giving here like this gene is an olfactory receptor. It's called OR51B4. And this gene is known to be overexpressed in HCT116 cell line, which is the cell line we're using in this experiment. So when we look at its perturbation effect, we see that it's affected cell organization. So cells are not as spread. But it's very difficult as a human if we have 18,000 conditions to really judge what the function of this gene based on looking at what's its effect visually. So what basically KCML gave us is that overlooking at all the features and incorporating gene ontology now, it can give us certain predictions what possibly this gene can do. So for example, it's predicted a role in negative regulation of epithelial cell proliferation, ion channel binding, as well as mesoderm development. And this is very useful because this olfactory receptor, we don't know what its function beyond the nose. And actually, we have 400 olfactory receptors, and we know their role in sensing the smell, but they also expressed in other tissue types, and we don't have sufficient knowledge what they do. So this is like KCML can already give the biologists some hypotheses that they can test. And from this analysis, what something interesting came up is that actually these are the genes involved, predicted to be involved in mesoderm development, so very dense spaghetti network of interactions. But what really catched my eye is that actually 83 olfactory receptors were predicted to play a role in mesoderm development. So I thought to follow up on it a bit more. And what other, like here, I'm showing some other examples that were also predicted to play a role in mesoderm development. So we have genes involved in TGF-beta signaling and integrin signaling, as well as Wnt signaling. And this is very interesting because most of the genes that came up in this analysis and associated with this function were associated with or enriched for colorectal cancer pathways. So it seems like they really might have a role in cancer progression. And just to explain what mesoderm development for people who might not know is basically when in development, when cells break the basement membrane and start migrating to form a new specialized layer. And this is basically what we observing in these images. In these images, cells are, when we perturb these genes, cells are clumped and not able to separate to form an epithelial layer. So I wanted to validate this a bit more, and I'm not getting a lot into the details, but just to give you a big picture. So I used gene expression data from colorectal cancer patients. And you can here see like the different molecular subtypes based on all genes in BCA space. So if I used the top predicted mesoderm development genes, so around 300, it also recapitulate these molecular subtypes, indicating that these genes are relevant to cancer progression. I also found that actually mesoderm genes and olfactory receptors are correlated with TGF-beta and Wnt signaling in patients, which confirm what I found in vitro, that perturbing these gene sets also result in similar phenotypes. And here you see also like by looking at the combination of olfactory receptors as a metagene, that expression of olfactory receptors in grade 3 patients predict worse survival. So all these suggest that actually we can identify genes involved in multicellular organization in vitro, and these are biologically relevant and can help us identify their role in patients. So from this, was the question how we can identify how we can systematically validate KCML predictions. So I used annotations that added over 18 months to gene ontology database. And I found that actually many of KCML predictions can be validated using based on the new annotation. So of course, KCML can be applied to image-based data, but also to CRISPR screens measuring viability, or siRNA screening measuring gene expression data. So basically, what's just says that KCML can augment existing gene ontologies to generate cell type specific gene annotations. So in summary, for this part, using prior knowledge is an important step toward harnessing the power of large imaging data. And in this case, I used prior knowledge on gene functions to guide phenotypic discovery and associate new genes and predict gene function. So at the end, I would like to give you an update on Phenoplot, which is a tool that I developed for visualizing microscopy data. So basically what here we see that we acquire a lot of images, and then we try to measure the shape features or different features and cluster these cells or cluster different breast cancer cell lines. However, when we represent, for example, in a heat map, the variable values as a color, or in bar chart, the variable value as the length of the bar, it doesn't help us to understand how this data look like in relation to the original images we measured. So to tackle this issue, I developed Phenoplot, which provide a completely new way, so new graphical elements that allow us to map our measurements to something that look like what we measured in the first place. So here is like a automated way that we map the thousands of measurements to thousands of cells to generate pictorial representation of these imaging data. And basically what it do is define a graphical element that, for example, for a cell, we can represent it as an ellipse, and the dimension of the ellipse can be represented based on our features. Same for the nucleus. We can represent the color using, or the protein abundance using a heat map fashion, like in a heat map like fashion, we can color the different ellipses. We can also represent cell-to-cell adhesion based on how the extent the thick line go around the ellipse, or the jagged line go around the ellipse can also represent irregularity of cell border. We can fill the ellipse with symbols depending on the variable value, so texture or number of organelles can be represented in this way. So basically, by applying Phenoplot to these measurements from breast cancer cell lines, we can have a much nicer way to represent these data, to interpret their meaning. So we see that cluster 2, which have luminal cell lines, have this small cells, not protrusive, does like have more regular border and more cell-to-cell adhesion. While cluster 3 and 4 is much more separate. Cluster 3 have more adhesion than cluster 4, while cluster 4 is more roughly. And what this gives us more intuitive way to look at the data and understand what these different phenotypes based on large number of measurements mean. So now we developed this the original version was in MATLAB. So now we developed a web-based app that's still not publicly available. We hope that we can release it this month. And basically you can upload your data, you can add multiple objects, and depending on the shape, you can choose the shape, and then you can customize the features you want to map to these objects. So now I will, just a simple example, we have the Metabrick data set, which measured genomics feature from 2,000 breast cancer patients, and it was clustered based on the genomic and transcriptomic features. And in that paper, they have these features like tumor size, number of lymph nodes, grade, and cellularity. So I average this for each cluster. And here is a demo. Hopefully it will work. So basically you load the data. So these data I just showed in the table, and then you create an object. So the tumor is represented by a circle. You specify the size based on the tumor size. And then the grade is assigned based on the color, so you can map it in like this gradient of color. The number of dots here represent the cellularity, and you also can customize the color. And the number of dashes that will appear now is basically represent how much the tumor is spreading to the lymph nodes. We can organize this in a scatter plot, so you are not losing your scatter plot power. So for example, the position of each glyph or phenoplot is based on tumor size and lymph nodes. And here, for example, you can see that cluster 9 showing tumors that have high grade, high cellularity, and spreading to the lymph nodes, while cluster 10 has high cellularity, but not spread to lymph nodes and lower grade. So here, just another example using wound healing data. We can put the wound height as well as the density of the cells. So of course, there is many examples we can give, but we're looking for nice demos. So if you are interested, if you have interesting data, or maybe we can think about designing additional features for your data, please get in touch. So in summary, topological metrics are important for characterizing tissue-level phenotypes in endothelial networks and scratch assays. KCML also provides scalable approach for analyzing large-scale genetic screens and inferring gene functions by incorporating prior knowledge. And lastly, like the last technology is also by visualizing data sets effectively, we can facilitate the interpretation. So it's basically developing different technologies that allow us making sense of complex imaging data. So at the end, I would like to thank my funders, the Wellcome Trust, for awarding me the Sir Henry Wellcome Fellowship, which was fantastic opportunities to visit and work with different laboratories. And the study on genetic screens is in collaboration with Lucas Belkmans. The study on endothelial networks is with collaboration with Ayman Zain, and the wound healing study with Jens Richer. Thank you for listening, and I'm happy to take any questions.
Thank you very much. Such beautiful images as well. So let's see. Do we want to do the Mentimeter maybe first?
Yeah, if anyone has any questions, please do put them into the chat, and then we'll go through them after that, I guess.
I'm willing to try my Mentimeter luck first, if you kind of wish. So I put the Mentimeter already into the chat. I think people are already responding. So I'm sharing my screen. I think it's this one here. I'll try my luck one more time. You're laughing. So there we go. This is your, what are the greatest challenges for your imaging experiments? I was just trying to figure out, people say high volume. Oh, I'm the kind of greatest challenge. Oh, that's very nice. Embedding, image analysis, image segmentation, sample health. Yes, maybe I like that I am the greatest challenge. I wonder who put that there. Oh, okay. So let's start the quiz. So here, this is actually the real quiz. So, well, not many people have joined then. I should wait actually until more people have joined. Come on, come in. Come on, guys, more people join. Come into my Mentimeter. Ching, ching. Strawberry, Santa Claus. Come on, you can do it. So at the end of the quiz, you know, you will be given a beautiful foldscope if you are fast and curious. We don't have many people today. Maybe on the Mentimeter show. Come on, one more. Yes. Yeah. All right. So it's always a bit daunting starting this because you can't really see people. Okay, I'm going now. This is your last chance. All right. Yeah, not to delay this much. We got some very nice questions referring to the talk. KCML is a tool for analyzing genetic screens measuring. And that was very nice because I heard the answer actually in the introduction by Nick. Your choices: cell shape, gene expression, viability, or all of the above. The answer is all of the above. I mean, that's why it's so powerful. And we go to the next question. Question 2. How many olfactory receptors do humans have? How many olfactory receptors help you sense? 400, 200, 700? Again, I think this is a question answer which you should have picked up. So 200 was not enough, 700 was too many, 400 is the right answer. Now the next one. Oh, sorry, someone was asking how you join. You click on the link in the Mentimeter for next time. So question 3. So I think it's now too late, but bar charts represent numerical data using color, length, position. I like this one. This is nice. This is very, it's a bit cheeky, really, I guess. Well, not really, but the answer is time is up, length. And only 3 people got a little bit wrong. Then here we have question 4, which is Phenoplot is a tool for, and I think you should get this right, because I mean, Heba has obviously developed this very cool analysis tool. And well, the answer is image analysis, data visualization, or clustering. And it's data visualization. Great. And now the final question, number 5, is Phenoplot web-based can represent only cell shape data, only tissue data, any phenotypic data. Okay, excellent. So everybody got the last one right, which is wonderful. So I think that means they got your talk. All right, so the winner, I think, is the little snowman Elenitsa. You were the fastest and you had the most answers right. So like in Mentimeter tradition, you need to kind of put your email and send it to me in the chat, or email me to my email, which will remain sr411@cam.ac.uk, but just put it in the chat. And over to the question and answer now. So I stop sharing. Well done. Thank you.
Right.
Okay, back to the questions. Shall
I take those questions?
Yeah.
We've actually got any in the chat at the moment. Good look at that. Oh, yeah, awesome. Okay, we have a question from Pablo Oral Vallis. Just wondering, how do certain difficult cell types result in decreased measurement accuracy? If so, how do you deal with less than standard accurate measurements?
Yeah, that's a great question. I think we always have this like compromise when we're doing analysis methods, that you try to adjust the method too much, but doesn't work for all images. So one thing I noticed in practice, that if you measure so many aspects, like the texture features, the segmentation, etc., the morphology, then some features can compensate for each other. So even if you have like not very accurate measure, if all the measurements will be different because of the segmentation, for example, error, then they will have different signature from a cell that is segmented correctly. So somehow, like, I find like having more measures that represent as much as possible of what we're seeing sometimes can circumvent this. But still, like, it's always difficult if you're trying to develop one metric that capture one biological feature. Is always difficult, like, to get it perfect. So it's about like addressing the segmentation challenges to make the feature more robust.
Right. Actually, can I, since I'm talking, can I just ask you a bit more on that? So if you go back to your sort of triangle of the genes, the function and the phenotype, I think in the middle part, you talked about the cell adhesion assay, and I think you briefly mentioned the too many phenotypes were sort of overwhelming your ability to cluster the specificity. Is that right? Or did I miss that point?
Yeah, exactly. Like, I think often when we do these experiments, there is a lot of heterogeneity in the cell phenotype, the resulting phenotype. So if you, like, for example, the number of cells that will be mitotic if you knock down a cell cycle gene will not be like it often will be a small percentage. And therefore we need like really powerful methods that can extract the signal, like even if there is a slight change in specific subset of features, that can make sense of these signatures as long as it's consistent, and then use this to improve the predictivity of gene functions. Does that answer your question?
Well, I guess I sort of what I was asking, is there a human element to how you sort of prune the inputs, or do you?
Yeah, I think usually people who work with these data sets, there is a lot of human elements all through the process. So I think with KCML, I try to take out the human elements because it is really difficult then to compare results from one study to another. So I think what I'm trying to do is to eliminate how much human will be involved in defining and crafting these features because to make the analysis more systematic.
Okay. We've got a question here from Lucia Pinon Giraldes. What platform was used to acquire the images? I mean, I get you talked about a few different screens there, so maybe you could just say that.
Yeah, exactly. So the breast cancer cell lines were acquired in from my PhD lab. So it was Berken, Elmar, Operata. But then the other ones that the genome-wide screen is the Yokogawa system. So it's all like high throughput microscopy systems.
Right. And what about the wound assay one?
I think that one was wide field microscopy, but it's not it's data available publicly. I didn't acquire the data, so.
Right. Okay.
You can also unmute if someone wants to ask a question specifically. But I think, no, it's okay. It's always a bit daunting, I know. To see anymore. But yeah.
All right, well, I'm going to pick on you, Alessandra. You have any questions you were thinking about asking there, Alessandro?
I mean, for me, it's always interesting to understand, you know, also what next from the point of view of the imaging acquisition. So now, I mean, in the past, I've been involved in the development of, for instance, high throughput film systems, but fluorescence lifetime imaging microscopy in high throughput context didn't really take, you know, didn't really become a common thing. Is there something from the imaging perspective that you think that could actually have an impact in for this type of data? So which type of features you think would be interesting to add?
From the imaging perspective?
Yes.
I think maybe I think the effect is more the other way around. So you said you were involved in high throughput film, but it wasn't adopted. I think the reason it wasn't adopted is usually people will find the analysis challenging and they will step back and say, okay, we can't make sense of this data. So I think tackling the challenges of analysis maybe will make it easier, like to generate these data sets. Like I think also with in terms of 3D imaging of spheroids, also people find it also very challenging to analyze.
Yeah.
From imaging perspective, I think for us is mostly like 3D imaging would be something interesting, and how we can capture good quality 3D imaging data of spheroids, of organoids. Like we try to work on this project, but however, it was really limiting what we can acquire with imaging to be able to analyze it really in to make sense of the resulting features. So
Yeah.
I think they both impact each other. Yeah.
Yeah. No. Absolutely. Yeah. Thank you.
Okay. Well, I have one last question. I mean, these are very, very large data sets. How sort of computational time is it taking to get a sort of an output out of these systems?
Well, I think, yeah, it is. Usually we run things on the cluster. So, and especially like with the graphical GPUs and the graphical processing units, deep learning, like it will take, so once you train the models for learning, it's like between like few days, but sometimes a month or something like that. But I think in terms of computational power, we're still managing. I guess with tissue imaging data, it's becoming more complex because people are acquiring so many markers. So yeah, I think in terms of these data sets, still because it's few markers, it's still like it's okay. Like I think we can handle it fine.
Right. Okay. Sure. Okay. In that case, if I don't think we're getting any other questions, so maybe we could just give our speaker, Heba,
Round of applause.
round of applause. And thank you very much.
Next time we give you hopefully lunch and kind of teas and coffees and cakes.
Yeah.
Thank you very much. It was
Thank you very
really, really
Thank you.
nice. Thank you.
Bye.
Very inspirational and really relevant. Thank you.
Bye.
Thank you.
Bye.
Bye.